Hello, my name is Professor Hudson. You are about to view a narrated PowerPoint lecture on Chapter 5, Cell Membrane Structure and Function. The topics to be covered include 5.1, how is the structure of the cell membrane related to its function, 5.2, how do substances move across membranes, and 5.3, how do specialized junctions allow cells to connect and communicate. Let us begin. All the membranes of a cell have a similar basic structure. This structure includes proteins suspended in a double layer of phospholipids. Proteins are responsible for selectivity in exchanging substances and communicating with the environment, controlling biochemical reactions, and forming attachments. This slide shows the plasma membrane. Functions of the plasma membrane include: it isolates the cell's contents from the external environment, it regulates the exchange of essential substances, it allows communication between cells, it creates attachments within and between cells, it regulates biochemical reactions. Water-soluble substances such as salts, amino acids, and sugars cannot easily cross phospholipid bilayers. However, the isolation provided by the plasma membrane is not complete, and very small molecules such as water, oxygen, and carbon dioxide, as well as larger lipid-soluble molecules, can pass through this selective barrier. This slide shows what a phospholipid looks like. The membrane of your cells is made up of phospholipids. Phospholipids are the basis of membrane structure and consist of two very different parts: a polar hydrophilic head, which is illustrated by the brown circle, and two nonpolar hydrophobic tails, which is illustrated in green. Remember, hydrophilic means love water. Hydrophilic substances mix with water. Hydrophobic means fear of water. Hydrophobic substances do not mix with water. So the phospholipid has a hydrophilic portion and a hydrophobic portion. The outer surface of animal plasma membranes are bathed in a watery interstitial fluid, which is a weakly salty solution resembling blood without its cells or proteins. Membranes are sometimes referred to as fluid mosaics. The fluid mosaic model of a membrane was proposed by Singer and Nicholson. This model illustrates that each membrane consists of a mosaic or patchwork of different proteins that constantly shift and flow within a viscous fluid formed by a double layer of phospholipids. A fluid is any substance whose molecules can flow past one another and include gases, liquids, and cell membranes. The fluid phospholipid bilayer helps to isolate the cell's contents. The phospholipid bilayer is the fluid portion of the membrane. Plasma membranes face both the exterior, outside of the cell, and the interior, watery cytoplasmic environment. Here is another snapshot of the phospholipid bilayer. Notice it is two layers of phospholipids. The inner portion of the phospholipid bilayer consists of the hydrophobic tails, and then the hydrophilic heads face either into the cytoplasm or outside the cell. The phospholipid bilayer is flexible and fluid, which allows for cellular shape changes. The individual phospholipid molecules are not bonded to one another. Some of the phospholipids have unsaturated fatty acids, which causes their tails to form kinks. Membranes become more fluid at high temperatures and less fluid at low temperatures. Cell membranes of organisms that must live at low temperatures tend to be more unsaturated. The kinks help the membrane stay fluid in what would be normally an environment that would make the membrane want to become less fluid. Cholesterol stabilizes membranes and affects fluidity and reducing permeability. So cholesterol is important because it allows the membrane to be fluid and move around, but at the same time it prevents some molecules from passing. Let's review. Question 11. The plasma membrane is composed primarily of, choose the correct answer. This slide is showing another view of the phospholipid bilayer with its embedded glycoproteins. It's also showing the types of molecules that can enter the cell and which ones cannot enter without assistance. Small charged molecules and ions typically cannot pass through the phospholipid bilayer without assistance. Water can enter the cell using specialized channel proteins called aquaporins. Non-charged molecules can easily pass through the phospholipids, whereas large macromolecules need assistance to enter the cell. Gases typically enter the cell very easily by simply diffusing across the phospholipid bilayer. Let us review. Question 12. Which of the following does not move freely without energy or a carrier protein across the plasma membrane? Choose the correct answer. A variety of proteins form a mosaic within the membrane. Proteins are embedded within or attached to the phospholipid bilayer. Many proteins have attached carbohydrates on their outer membrane surface. We refer to these as glycoproteins. Glycoproteins play a role in cell recognition and allow the body to recognize self from non-self. If you were to get an organ transplant, the glycoprotein profile on the organ you receive would have to closely match that of your body, or else your body would attack it. We call that rejection. Even if the glycoprotein profile matches very closely, you still would need to take immunosuppressive drugs to keep your immune system from attacking the organ that was transplanted into your body. This slide illustrates some of the other functions associated with the proteins found in the plasma membrane of the cells. Membrane proteins may be grouped into 5 categories. Enzymes, recognition proteins, is what I just mentioned, receptor proteins, connection proteins, and transport proteins. Please read the descriptions in your book or on this slide for each one of the categories discussed above, so that you will know what each one of these categories refers to. Now we're going to switch gears a little bit and talk more specifically about the processes that allow substances to move across the plasma membrane. This is covered in section 5.2 of your textbook. So how do substances move across the membrane? Some substances can move across membranes by diffusing through phospholipid bilayer or through specialized transport proteins. Before we talk more about the processes associated with moving molecules across the plasma membrane, let's look at some definitions relevant to substance movement. A solute is a substance that can be dissolved or dispersed, such as atoms, ions, or molecules, in a solvent. A solvent is a fluid capable of dissolving a solute. Water is often called a universal solvent because it dissolves a lot of different types of solutes. When a solute is dissolved in a solvent, the resulting combination is called a solution. The concentration of a substance defines the amount of solute in a given amount of solvent. A gradient is a physical difference in temperature, pressure, charge, or concentration of a particular substance in a fluid between two adjoining regions of space. Cells use energy-requiring transport proteins to generate and maintain concentration gradients, the differences in solute concentration across their membranes. Now that we have defined some terms, let's discuss how molecules move in general, and then discuss how they move in and out of cells across the cell membrane. In this slide, we have a beaker filled with what we call a solution. The red dye molecules represent the solute. The blue molecules represent the solvent, which is in this case water. When a drop of dye, which is the solute, is placed in water, initially they are concentrated in one spot. But because molecules move from where they're more concentrated to where they're more spread out, or where they're at a lower concentration, eventually these red dye molecules will spread out throughout the entire beaker. As you see in the second image, the dye molecules, which is the solute, diffuse into the water, and the water molecules diffuse into the dye. Both dye molecules and water molecules are evenly dispersed. An example of this in everyday life would be if you were to make Kool-Aid. The Kool-Aid powder would represent the solute, and the water would represent the solvent. When you initially pour the Kool-Aid powder into the pitcher of water, the Kool-Aid powder will be concentrated in one location. But over time, the Kool-Aid powder solute particles would move throughout the water, and the water molecules would move throughout the solute Kool-Aid powder until the pitcher contained evenly dispersed Kool-Aid powder and water. One process we use to speed this up would be to stir. If you were to stir the Kool-Aid while you were making it, this process of diffusion would occur much quicker. Molecules have internal kinetic energy that causes them to move. Diffusion is when molecules move from high to low concentration until equilibrium is reached. In general, diffusion can happen in a semi-solid liquid or a gas. The above shows diffusion in a liquid. Diffusion is the movement of solutes from regions of higher concentration to regions of lower concentration.